Welcome back, guys. Today we are discussing cardiac and smooth muscles in muscular physiology. Beforehand, we have already discussed skeletal muscles, so go check that out if you haven't. So let's start with this. Let's first start our discussion with cardiac muscles. Okay, first is cardiac muscles. Okay, cardiac muscles are present in heart exclusively. We know this fact already. Exclusively present in heart, the cardiac muscles. Okay, now the contraction process is involuntary. Contraction process is involuntary, that is with the help of pacemaker. What is the dominant pacemaker here? SA node. These are the already basic facts we know here, that the contraction process is involuntary with the help of pacemaker SA node. Okay, composed of actin and myosin. Okay, now the type of myosin here is different. Okay, myosin, here we have only heavy chain in heart. Okay, and this heavy chain in heart has two subunits. So first is alpha subunit. Alpha A for A is present in atrium. Okay, and other subunit is beta. Okay, this is present in ventricles. A for A, alpha atrium, B in ventricles. Remember, only heavy chain. Okay, along with actin and myosin, these are the contractile proteins. Okay, now the regulatory proteins are tropomyosin and troponin, both are present. Tropomyosin and troponin, both are present. Okay, after the regulating proteins, another protein, the titin, the spring-like structure, spring-like protein, and the largest protein and body is also present here. Okay, this is elastic in nature. Okay, elastic in nature. And what is this function? On stretching of cardiac muscle. Okay, on stretching of cardiac muscle, it leads to recoil. Okay, this is called elasticity. Okay, these are elastic recoil. Okay, this is due to titin. Now this mutation in titin in heart, mutation in titin protein in heart, no recoil on stretching. Very simple funda, no recoil on stretching, which leads to dilated cardiomyopathy. Okay, this condition prevails. Dilated cardiomyopathy due to mutation in the titin protein, where in the heart is responsible for elastic recoil. Okay, there is no recoil on the mutation. Okay, so this was the titin protein. Okay, now we'll see a characteristic feature of the cells of the heart. Okay, so first we'll draw these some cells. Okay, this is the first cell, this is the second cell, the third cell, and it goes on and on. Okay, goes on and on with the last cell. Okay, in a row, this is the first cell, this is the last cell. Okay, we know that in between these cells is present of different junctions. These junctions, what are these junctions? These are our gap junctions. Yes, gap junctions. Okay, now we'll write that impulse travels from first cell to last cell, okay, very quickly, very quickly, okay, so that all can contract together, all can contract together at the same time, at the same time, okay. This is called syncytium, 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 functional syncytium, functional syncytium, okay, is due to this gap junction. There is this gap junction in the between the myocardial cells. Once gap junction requires connexin proteins, sorry, connexin, write it again, connexin proteins. Important question asked frequently, present at intercalated disc regions, intercalated disc regions requires connexin protein. Connexin protein is required for gap junctions for functional syncytium, so that all can contract together at the same time. In the heart musculature, all of the musculature contracts together all at the same time, because due to this gap junction, which is due to our connexin protein, connection proteins. Okay. Now we'll see some unique features of cardiac musculature. Unique features. Okay. We'll expand our discussion on this. The first is our length tension relationship. Length tension relationship. Okay. So we have our length factor. Okay. Now this length factor is changed by blood volume. Changed by blood volume. It is our end diastolic volume. We'll learn everything in the cardiovascular physiology. Don't worry. Just remember, it is changed by the blood volume, which is our end diastolic volume. Now this end diastolic volume, it stretches the chambers. Stretches the chambers. This is the length factor. Now there is the tension factor, which one is due to force. This is due to force of contraction. Force of contraction. That is pressure in left ventricles. Pressure in left ventricles. Okay. Now if we draw a graph, in the y-axis we take pressure. It is our tension. Okay. And on the x-axis we have our length. Is our end diastolic volume. End diastolic volume represents the length. Okay, this is the x-axis, this is y-axis. If we draw the graph, this will come out like this, somewhat like this. Okay, so this suggests that this will lead to conclusion that the force of contraction is directly proportional to initial length of muscle fiber. Force of contraction is proportional to initial length of the muscle fiber. What is this force contraction? Pressure factor. What is this length of initial length of muscle fiber? Represented by end diastolic volume. This is called our Frank-Starling's law. This is our Frank-Starling's law. The force of contraction is all directly proportional to initial length of the muscle fibers. Now the additional point to be added here, that this law only acts within physiological limits. Very important point. Within physiological limits, the force of contraction is proportional to initial length of muscle fiber. As the length increases here, up here, okay, contraction it's proportionally increased up to a certain point. Okay, up to a certain point. Now, this region is within physiological limits. Okay, after which, on increasing further length, the tensional pressure decreases. Okay, this is our Frank-Starling's law, which acts within physiological limits. Okay, yes. So this is the first unique feature on length tension relationship, which gave us our Frank-Starling's law. Moving on to the second point, we have CICR. What is the CICR? Calcium induced calcium release. We have CICR, which translates to calcium induced calcium release. Calcium is used to induce the further calcium release. Okay, now we'll see what happens here. Suppose this is a myocardial cell in the heart. Okay, now this is our sarcoplasmic reticulum. Okay, sarcoplasmic reticulum, which obviously contains our calcium ions. Okay, as calcium is released outside, okay. For this release, extracellular calcium is needed for this release. Extracellular calcium is needed for this release. Okay, so an important take home point here in heart. Okay, both extracellular and intracellular calcium are needed. Okay, both extracellular and intracellular calcium are needed. But in skeletal muscle contraction, we have seen that only intracellular calcium from the sarcoplasmic reticulum is needed. Okay, from sarcoplasmic reticulum only extracellular calcium is needed. Okay, yes. So now we go on to our third point. Okay, third point here, there is the action of this norepinephrine. Okay, norepinephrine. Okay, now norepinephrine, this action, first we will see the what occurs in this cell. The cell has this obviously sarcoplasmic reticulum, which contains our calcium, where in the sarcoplasmic reticulum, terminal systems. Okay, now we have here our Sarca pump for relaxation of muscle. We have already discussed this in the skeletal muscle discussion. Go check that out. We have our Sarca pump. Okay, now there is this compound called phospholamban. Okay, this phospholamban inhibits Sarca pump and interferes with calcium exchange. We know ryanodine receptor is responsible for exclusion of calcium coming out of sarcoplasmic reticulum, and Sarca pump is for taking inside the calcium that has come out. But this phospholamban inhibits this Sarca pump. Okay, and this norepinephrine does what? It does phosphorylation of phospholamban, so that it is inhibited. Phospholamban is inhibited and in the Sarka pumps gets non-inhibited and functions properly. Now this is called positive lusitropic effect. Positive lusitropic effect of norepinephrine. Positive lusitropic effect of norepinephrine. So norepinephrine does what? Phosphorylation of phospholamine. Phospholamine was inhibiting the Sarka pump. So in turn the phospholamine is inhibited and calcium exchange occurs normally. Positive lusitropic effect of norepinephrine. Remember norepinephrine. Okay. So now this norepinephrine, okay, norepinephrine ensures adequate calcium storage. Norepinephrine ensures adequate calcium storage required for future contractions, okay, called positive ionotropic effect. What is this ionotropic effect? We will discuss everything in details in cardiovascular physiology. Don't worry. Just remember that is the contraction. Contraction is an ionotropic effect, and norepinephrine has positive ionotropic effect. Also positive lusitropic effect. Okay, inhibits the phospholamine, which was inhibiting the Sarka pump. Okay. Now a common question is asked everywhere that why cardiac muscles cannot be titanized. Why cardiac muscles cannot be titanized? Okay, so we'll find out the answer to this question here. Okay, so we have two events for the contraction of cardiac muscle. Okay, first we have our electrical event, which is our action potential, and then we have our mechanical event, which is our muscle contraction. Now the point to be noted here is that the electrical event action potential is different in cardiac musculature than it is in skeletal musculature. The action potential phase is different. Why different? We will discuss it in cardiovascular physiology, but remember that the pacemaker potential generation is different from the action potential generation in the skeletal muscle, where there is no pacemaker. So we have a pacemaker in the heart, the SA node. Its action potential general graph is a bit different. So we'll see only the graph. We'll discuss it in details in cardiovascular physiology. Okay. So this is the graph here. This is the graph here. Okay. This is the action potential graph, and here this is the muscle contraction graph. So we now we have two refractory phases. Okay, refractory periods. One is our absolute refractory period and our relative refractory period. Okay, now this is our absolute refractory period. This is our relative refractory period. Okay, very important understanding. See what happens in the cardiac muscles. Okay, the major part of contraction, okay, lies in the absolute refractory period. Absolute refractory period. And we know in the absolute refractory period how much of a strong second stimulus is given, action potential will not be generated. So the second stimuli given here can't be submitted second stimuli can't be summated. So as a result, cardiac muscles can't be detonized. Okay, first we need to remember is that major part of contraction lies in ARP, absolute refractory period, and in absolute refractory period second stimuli can't be summated. Hence cardiac muscles can't be detonized. Okay, so here is the graph. Here the action potential event, and this is the muscle contraction event. So majority of this contraction phase here lies in the absolute refractory part, and here the second stimuli given how much strong, it doesn't matter, it can't be summated, and that is the reason why cardiac muscle can't be detonized. Okay, so this was our understanding. This was a total discussion on our cardiac muscles. Okay, moving on to our smooth muscles. Okay, second discussion is our smooth muscles. So in smooth muscles, some features will see that there are no striations. Okay, and the contraction is involuntary. We all know this. Contraction is involuntary, so also requires a pacemaker. Okay, requires a pacemaker. Now this pacemaker of smooth muscles is called Kajal cells. Kajal cells. Write this again. Kajal cells, the pacemaker of the smooth muscles. Okay, there is actin and myosin, but no Z lines. Okay, no Z lines, and instead of Z lines, instead have dense bodies. Instead of Z lines, we have dense bodies. Now some features of what proteins we have and what proteins we don't have. There is no troponin. Okay, instead have calcium binding protein. Okay, called calmodulin. It's a calcium binding protein instead of troponin. No troponin and no titin. So they are not elastic, which is no elastic recoil. Okay, so no troponin, no titin. Instead of troponin, calcium binding protein calmodulin, no titin, no elastic recoil. Okay, also no Z lines, instead dense bodies. What are the pacemaker here? Kajal cells, and we obviously know the very basic funda, no striations. Cardiac muscle do have striations, but they are also involuntary. Okay, now this was a brief introduction. Now we'll see types of smooth muscles. Okay, and these types of smooth muscles is on the basis of presence or absence of gap junctions. Absent the gap junctions in cardiac muscles mediated by connexin protein required for functional syncytium. This is are of two types. Okay, first is on the basis if it's present. Okay, these are called, now the gap junctions are present, these are called single unit smooth muscles. Single unit or unitary smooth muscles. Okay, very simple. These are present in GIT, gastrointestinal tract, in the uterus, in the bladder, and in the ureter, urinary bladder. Uterus, urinary bladder, ureter, and GIT, single unit or unitary smooth muscles with gap junctions. Now the gap junctions are absent. Now these are multi-unit smooth muscles. Okay, these are present in blood vessels. These vascular smooth muscles. Vascular smooth muscles are multi-unit smooth muscles. Let me write this once again. Multi-unit smooth muscles in blood vessels, that is our vascular smooth muscles. Okay, these are the types of smooth muscles based on presence or absence of gap junctions. Now we will expand our discussion to the two phases of our muscle, that is our contraction and relaxation. Okay, contraction and relaxation. We'll see what happens, what are the differences here. Okay, in the smooth muscles, first is our contraction. We have our here MLCK. What is this MLCK? Myosin light chain kinase. What do kinase proteins do? It's phosphorylation. Kinase proteins do phosphorylation. MLCK, myosin light chain kinase. Okay, this MLCK activation leads to contraction of smooth muscles. Okay, and the relaxation, we have MLCP, myosin light chain phosphatase. Myosin light chain phosphatase. This activation leads to relaxation of smooth muscles. Okay, so we have two things. So from this conclusion we came to the conclusion that both are myosin based regulation, contraction and relaxation. For contraction we have myosin light chain kinase, relaxation phosphatase. Activation leads to contraction of kinase, phosphatase activation leads to relaxation. Okay, now for this relaxation we have a next important point. Okay, this relaxation MLCP is inhibited by group of enzymes. Sorry, I'm writing very ugly here, I don't know why. Inhibited by group of enzymes. These enzymes group are called Rho kinases. Okay, they interfere with relaxation. Okay, now we have a Rho kinase inhibitor. There is a drug that inhibits the Rho kinase and in turn allows the relaxation to continue properly. This is our fasudil. this drug is a Rho kinase inhibitor. Okay, phosphatase. Okay, so the phosphatase inhibited Rho kinase. This Rho kinase inhibitor is a phosphatase, which will allow the smooth relaxation of smooth muscles without any further intervention. Okay, now moving on, we have the regulation of smooth muscles with ANS. Okay, regulation with autonomic nervous system. How autonomic nervous system exerts its effect with regulation, you will see here. Okay, so we have two phases, contraction and relaxation. Okay, and we have our first is our parasympathetic system here. Parasympathetic system. Okay, this causes excitatory potentials in smooth muscles. This leads to contraction. Parasympathetic is responsible for contraction in the part of ANS. Now, obviously for relaxation, we have our sympathetic system. And sympathetic system produces inhibitory signals. Inhibitory signals, which will lead to relaxation. Okay, sympathetic relaxation, inhibitory signals, parasympathetic system excitatory potentials, contraction, inhibitory signals or potentials. Okay, so sympathetic, parasympathetic, relaxation and contraction of smooth muscles. Okay, now in smooth muscles, okay, suppose this is a smooth muscle cell. Okay, so sarcoplasmic reticulum of the smooth muscle cell, and also we have a calcium here. Okay, we know ryanodine receptor responsible for the efflux of calcium. Ryanodine receptor. Okay, along with that, we also have our IP3 system, adenosine triphosphate receptor, which is also for efflux of calcium to outside. Okay, so this was the point, that along with ryanodine receptor, calcium IP3 receptor is also present where in sarcoplasmic reticulum. Okay, now we'll see the unique features of smooth muscles. Okay, the first feature is, we have already seen no tighten. Okay, no tighten, so no elastic recoil, on stretching, called plasticity. If it was elastic recoiling, it was called elasticity, but here it is called plasticity. For example, in uterus and urinary bladder, this plasticity occurs. Okay, due to no presence of titin. We also have seen there is no troponin and no Z lines. Okay, now the second unique feature, okay, in GIT. Okay, in GIT, smooth muscles, this can be maintained in a state of sustained contraction. Can be maintained in a state of sustained contraction, okay, with very little use of ATP. With a very little use of ATP, this can be maintained in a state of sustained contraction, and JIT smooth muscles. This is called our latch bridge phenomenon. Latch bridge phenomenon. We've seen a latch bridge, how once it starts vibrating with to and fro motions, it does not stop. It stops after a very long time. So the vibration of the bridge can be maintained for a very long time. So is this phenomenon here. The contraction, sustained contraction maintained for a very long time with the help of very little use of ATP. Very important point here. ATP is very less used. This is called our latch bridge phenomenon, seen in smooth muscles of GIT. Okay, and the other is no tighten here. So there occurs plasticity. Next is we are going to discuss the synaptic transmission. Okay, synaptic transmission. Very important topic for discussion. Let's get started. Now, there are two types of synaptic transmission. Okay, first is our electrical synapses, and chemical synapses. Chemical synapses. Okay, this electrical synapses is in gap junctions, and it's a fast process. Okay, and this chemical synapse is due to release of neurotransmitters, and it's a slower process. Despite being a slower process, this is the majority one in our biological human system. Okay, now we'll see a synapse. Okay, see a synapse. Synapse was given by Sir Charles Sherrington. Sir Charles Sherrington, father of synaptic transmission. Synaptic transmission. Okay, so we'll start with a neuron. Okay, neuronal cell. Cell body. Okay, and this is the axon. We have our axon terminal. Okay, now beyond this axon terminal, we have our synapse region. Synapse region. After the synapse region, we have our postsynaptic neuron. Okay, postsynaptic neuron. So let's expand our discussion here. This is a presynaptic neuron. Presynaptic neuron. Okay, this is our axon. So this is called our axosomatic synapse. Axosomatic synapse. Okay, most common type of synapse in CNS. This is the most common type of synapse in CNS. Most common type of synapse in CNS. So in this axon terminal, okay, we have our neurotransmitters in the vesicles. Neurotransmitters in the vesicles, such as acetylcholine, norepinephrine, epinephrine, and so on. It's not our discussion. Discuss further in the neurotransmitter section, but most common is our acetylcholine. Okay, because now we have discussed before that there is depolarization, okay, due to sodium influx. I'm discussing again depolarization. It goes from minus 70 millivolt to minus 40 millivolt. Minus 40 millivolt is the firing level for opening of calcium channels. Calcium influx leads to exocytosis of neurotransmitters. These neurotransmitters get exocytosed the vesicles and goes up to here and combines with their goes up to the synaptic region, okay, and generates action potential in the postsynaptic neuron. This is our postsynaptic neuron. Okay, this is our synapse region. Okay, so this was our brief understanding of synapse. Now, beyond the synapse, we have our postsynaptic neuron, where these action potentials are generated. Now, we'll see some different types of postsynaptic potentials. Postsynaptic potentials. Okay, postsynaptic potentials. Now, this is again of two types. Okay, first is our excitatory. First is our excitatory. This is due to glutamate neurotransmitter. And the other type is our inhibitory. This is your due to gamma aminobutyric acid or glycine. GABA or glycine, excitatory and inhibitory. Now this excitatory this is due to glutamate. This is called EPSP. Excitatory postsynaptic potential. Inhibitory IPSP. Inhibitory postsynaptic potential. Okay. Now this is again of two types. Excitatory fast postsynaptic potential is of two types: fast and slow. Okay, fast and slow. In fast PSP, the cell interior becomes less negative. Due to influx of sodium ion or calcium ion. Not only sodium ion, and other positive ions such as calcium can also do this. Due to influx of sodium or calcium, cell interior becomes less negative. Okay. Now slow PSP has higher latency of 500 milliseconds. That's why it is slow. This is due to decrease in potassium efflux. Okay, this is due to potassium efflux decrease. Positive ion influx is decreased, so the cell interior becomes less negative. The high latency of 500 milliseconds. So that is why it is slow. Also, this IPSP, inhibitory postsynaptic potential, is also of two types: fast and slow. In this first type, cell interiors become more negative. Cell interior becomes more negative. It is inhibitory, going towards hyperpolarization. Okay, due to a negatively charged ion inside. Okay, which could be due to chloride influx. Chloride can infuse, or this can be also due to potassium efflux. Fast potassium efflux. Potassium is effluxing, we know, will lead to hyperpolarization phase from repolarization and leading to hyperpolarization phase. It will be chloride influx or fast potassium efflux. This is fast IPSP. Now in this slow IPSP, the latency is also very high. Slow is also 500 milliseconds. 500 milliseconds, not meter per second, milliseconds. 500 milliseconds is the latency here, and this is due to increased potassium efflux. Increased due to potassium efflux. So I've seen, barring this first IPSP, everything is related to potassium efflux, either increased or decreased. Increased where? In IPSP, both fast and slow. Inhibitory. Inhibitory is always potassium efflux. Okay, and this is always fast. Only addition in the first IPSP is due to chloride influx, and slow EPSP is due to slow potassium efflux, decreased potassium efflux. Very important discussion here to be revised a lot of times. Okay, post-synaptic potentials are asked. Okay, now these slow potentials, particularly the slow potentials, both EPSP and IPSP, are seen in autonomic ganglia. Autonomic ganglia, also in smooth muscles and in cardiac muscles. Okay, autonomic ganglia, smooth muscles and cardiac muscles, which is slow potentials. Okay, now expanding our discussions on this, the inhibitory or slow potentials. Okay, this is due to potassium ions only, or due to potassium ions only, seen here, inhibitory and also the including slow potentials. Okay, now the EPSP, excitatory postsynaptic potential, is a type of local potential. It's a type of local potential, not an action potential. It's not an action potential. Okay, so in neuromuscular junction, EPSP can always be submitted. And can cross the threshold and action potential fires. EPSP can also always be submitted, and upon crossing the threshold it can fire. Okay, this was all about our discussion on EPSP and IPSP, the all types of post-synaptic potentials. Now moving on, the regulation of synaptic transmission. Regulation of synaptic transmission. Okay, the synaptic transmission is here regulated. Now this is again of two types. is either facilitated or inhibited. Facilitated or inhibited. Okay, so how is facilitated? Which ions facilitate? Calcium channels open for more duration. Open for more duration. There is enhanced calcium influx. Enhanced calcium influx, which is facilitatory. Calcium ion remains open for a long time, leading to enhanced calcium influx. And inhibited, how it's inhibited we'll see. So this is a presynaptic neuron, its axon and the end synaptic terminal and axonal terminal. Here we have our neurotransmitters. Now this neurotransmitter is a inhibitor neurotransmitter such as GABA, which is released. Okay, after releasing, it acts on a nearby neuron and inhibits it. So presynaptic, this is a postsynaptic. Okay, inhibited. Postsynaptic neuron is inhibited. This is called postsynaptic inhibition. Postsynaptic inhibition. The postsynaptic neuron is inhibited due to an inhibitory neurotransmitter such as GABA or glycine. We have seen this inhibitor is due to GABA or glycine, and excitatory is due to glutamate. Okay. Now moving on. This is our first type of inhibition. Okay. Now expanding this our inhibition, the second type. In our second type, we'll draw a neuron. Okay, this is our axon, this is our synapse. Okay, this is our postsynaptic neuron. This is our postsynaptic neuron. Okay, now there is a lateral neuron here. Laterally a neuron is present here, and this neuron inhibits the presynaptic neuron. This is postsynaptic. It's a lateral neuron from lateral direction. Okay, inhibits presynaptic neuron. Okay, this is called lateral inhibition. Special type of inhibition, lateral inhibition. This is seen in retina. Lateral inhibition seen in retina. A laterally placed neuron inhibits the presynaptic neuron, not postsynaptic, presynaptic neuron, and this is seen in our retina. And the third type of inhibition we are going to see here. Okay, regarding the alpha motor neuron. So this is our alpha motor neuron, which is axon. Alpha motor neuron. Okay, and this, and beyond this we have a special type of neuron. This is called Renshaw cell neuron. Renshaw cell neuron. Okay, now this alpha motor neuron discharges its action potential to the Renshaw cell neuron, and Renshaw cell neuron gets activated. But in turn, this Renshaw cell neuron on activation inhibits the alpha motor neuron. Alpha motor neuron discharges the action potential throughout the synapse to the Renshaw cell neuron. Renshaw cell neuron activates it. But upon this activation, Renshaw cell neuron, through its mechanism, inhibits the alpha motor neuron. Now this is commonly called feedback inhibition. Feedback inhibition. Restless neuron is feeding back. It's feedback inhibition. Okay. Now here Renshaw cell utilizes glycine. Inhibitory, either GABA or glycine. It uses glycine. This is needed. This feedback inhibition is needed for regulating or controlling the firing of alpha motor neurons. Regulating or controlling the firing of alpha motor neurons. The Renshaw cell inhibition is needed, which utilizes our glycine. Remember glycine to regulate or control the firing of alpha motor neuron. The Renshaw cell neuron inhibition of alpha motor neuron. Okay, so this concludes our total discussion on cardiac and smooth muscles. We discussed the proteins, the characteristics, the special properties, the different channels, also discussion on the synapse, the potential, the excitatory and inhibitory potentials, various types of them, fast and slow, which ions are involved, also the regulation, activation and inhibition. I discussed everything. Very important topic. Please keep revising them, guys. Thanks for watching. I'll catch you in the next one. Good luck.